It all began in a quiet laboratory in Japan, where a group of scientists set out to answer one of humanity's oldest questions. What makes us human, and how different were our ancient relatives? For centuries, the remains of Neanderthals, thick-browed, broad-chested cousins who once roamed Eurasia, had been locked away in museums and textbooks, their DNA preserved in ancient bones buried deep in caves. But now, through the miracles of modern science, researchers were ready to bring part of that ancient story back to life. At Kyoto Prefectural University of Medicine, two researchers, Dr. Ako Agata and Dr. Tadashi Nomura, stood at the threshold of history. Their laboratory hummed with the gentle whir of freezers and the sharp scent of ethanol, the air tense with the anticipation of discovery. Together with their team, they had managed to extract and sequence a strand of DNA more than 40,000 years old, a single gene variant that had once shaped the bodies of Neanderthals, long before modern humans walked the earth. They held in their hands not just a molecule, but a window into the past. The gene, known as GLI3, was thought to be responsible for critical steps in the formation of bones and organs during early development. It had been found in both Neanderthals and Denisovans, those shadowy relatives whose traces have all but vanished except for a few fossils and a genetic echo in modern people. But the researchers did not stop at simply reading the code. They wanted to see its effects in the flesh. Armed with CRISPR technology, a tool that lets scientists edit genes with the precision of a master calligrapher, they prepared to insert the ancient gene into living mice. The experiment was both daring and delicate, a bridge across 40 millennia, carrying a single mutation called R1537C. No one knew exactly what to expect. The mice were ordinary laboratory albinos, tiny creatures with pink noses and red eyes. Would the ancient gene change them? Would they suffer from the severe malformations that sometimes come with gene editing, or would something more subtle occur? Something that might answer the riddle of our shared origins. The first days were a waiting game. The altered embryos grew in the controlled warmth of their surrogate mothers, while the scientists watched, waited, and worried. As the pups were born, the team peered into their cages, noting every movement, every twitch of a whisker. At first, the newborn mice seemed much like their unaltered siblings, squirming, hairless, blind. But then, as days turned into weeks, changes began to appear. It was Dr. Nomura who first noticed it. He stood one morning before the clear plastic cages, his breath fogging the glass as he watched the young mice scurry in their bedding. "Come see," he called softly to Dr. Agata, who hurried over, notebook in hand. Several of the mice were different. Their skulls were broader, their ribs more widely spaced. When the team examined their skeletons under X-ray, they found even more surprises: elongated skulls, subtle curvatures of the spine, and rib cages that flared wider than any ordinary mouse's. The changes, though subtle, were unmistakable, echoes of the fossilized Neanderthal bones that lay thousands of miles and thousands of years away. The room filled with a hush, as if everyone was suddenly aware that they had crossed a boundary between the present and the ancient past. "These traits," murmured Dr. Agata, "they are exactly what we see in the old Neanderthal skeletons." There was awe in her voice, and more than a little fear. The mice had become living fossils, carrying inside them a trace of a lost humanity. As the weeks went by, the scientists kept careful watch. Some of the mice displayed broad, flattened crania. Others developed small curvatures in their spines, a gentle twist that brought to mind the strong, robust torsos of the ancient Neanderthal hunters. Even the mice's ribs seemed more twisted and arched, mimicking the fossilized skeletons that researchers had painstakingly unearthed from European caves. And yet, for all these profound changes, the mice were otherwise healthy. Unlike other experiments with gene mutations, there were no signs of missing limbs, no tragic deformities. The ancient gene seemed to work its magic with a subtle hand, reshaping the mice in ways that were clear to a trained scientist but did not destroy their health or vitality. The news spread through the scientific world like wildfire. In conference halls and university offices, researchers whispered about the Japanese team's findings. What did it mean for our understanding of human evolution? If a single gene, dormant for 40,000 years, could so powerfully alter the skeleton of a modern mammal, how many other secrets might our DNA still hold? One revelation struck the researchers with particular force. The mutation they had used, the R1537C variant, was not unique to Neanderthals. It still lurked, silent and rare, in the DNA of modern humans. It was most commonly found in people of European descent, present in anywhere from 4 to 8% of the population. In African populations, it was even rarer, but not entirely absent. This meant that the Neanderthals had not vanished without a trace. Their legacy still echoed in the bones and blood of living people. For the scientists, the implications were thrilling and a little unsettling. Could this be why some people today have broader rib cages or unusual bone structures? Might this ancient inheritance shape not only our appearance, but our susceptibility to certain diseases or conditions? As the team pored over data from the 1,000 Genomes Project, they saw patterns that suggested just that. The past, it seemed, was not as far away as anyone had thought. In the lab, the team watched the mice as they grew, documenting every change, every difference from their unaltered peers. They measured cranial widths, compared spinal curves, and even examined the shapes of the mice's tiny limbs. With every data point, the picture became clearer. The Neanderthal gene was a master sculptor, carving subtle differences that could, over many generations, define an entire species. Dr. Nomura, in a rare moment of reflection, remarked to his colleagues, "We are not so far from our ancient cousins as we like to believe. A twist in a single gene, a small difference in the script, and the story of humanity changes course." But the story was not only about the past. The team also wondered about the future. If such ancient variants could be reawakened and studied, might we also find clues to help humanity in the present? Could knowledge of these genes shed light on modern health challenges, such as bone disorders or congenital conditions? Might understanding our evolutionary inheritance help doctors develop new treatments or preventive measures? The researchers also discovered that the ancient variant, while powerful, was not destructive in isolation. In the wild Neanderthal population, relaxed developmental constraints may have allowed such changes to persist, giving rise to the robust skeletons that set them apart from modern humans. In mice, the genes shaped the body, but did not destroy it. It was a lesson in both the power and the limits of genetic inheritance. There was, of course, some concern. Not everyone was comfortable with the idea of resurrecting ancient genes in living animals. Critics questioned the ethics, wondering what unforeseen consequences might arise. But for the Japanese scientists, the rewards outweighed the risks. Their work was not about creating monsters or blurring the boundaries between species. It was about understanding who we are and where we came from, a journey as old as humanity itself. In the months that followed, the team published their findings. The scientific world buzzed with excitement, journalists and fellow researchers clamoring to know more. Some called the mice living Neanderthals, though in truth they were nothing so dramatic. They were simply mice, carrying a single ancient secret in their bones. Yet, the larger story remained. The Neanderthals, long thought to be gone forever, had left behind more than stones and tools. In the genes of living people, and now in the skeletons of these small red-eyed mice, their legacy endured, a quiet reminder that the past is never truly dead, only waiting to be rediscovered. And as Dr. Agata watched the mice in their cages, she could not help but feel a surge of wonder. Perhaps, she mused, the history of our species is written not just in books and bones, but in every cell, every twist of the DNA spiral. By looking to the past, we find the keys to the future. In the end, the mice grew old, as all creatures must. The scientists continued their work, delving deeper into the mysteries of human evolution, searching for the next hidden chapter in the story of life on Earth. And somewhere, deep in the hearts of those who listened, the tale of the Neanderthal mice was whispered, a story of curiosity, courage, and the enduring bond between the living and the lost.